Hello, so lesson objectives for today then. Discuss the fact that classification systems were originally based on observable features, but we're going to be looking at the more recent approaches which tend to be based on this molecular evidence. And then we're going to be looking at the 5-kingdom versus the 3-domain classification system. So last lesson you'd have talked about the 8 taxa, domain, kingdom, phylum, class, order, family, genus and species. Mnemonic I like to use for that one is Dapper King Philip came over for great soup. But there are other ones you can use. One that you do need to remember is the classification for humans. So they are in the eukaryote domain. They are in the Animalia kingdom, the Chordata phylum, the Mammalia class, primate order, Hominidae family, the Homo genus and the Sapiens species. And one thing that's worth remembering is that all mammals will have the same first 4 taxa there. So domain was going to be eukaryotes, kingdom Animalia, phylum Chordata, class Mammalia. With regard to the phyla, you don't need to remember the name of all of the different phyla because there are lots of different phyla, but I would remember Chordata and Arthropoda in particular. So Chordata are the ones that have a backbone or central nervous system. So majority of them are vertebrates, and you've got Arthropoda, which are the ones that have got a hard exoskeleton and jointed limbs. Types of things that you might get asked to do, so you might get given a phylogenetic tree like this one, and then you've got to try and classify a particular species. So this one we're going to have a look at the cat species. So domain is going to be the same as us, so it'll be Eukaryota, the kingdom will be Animalia, the phylum will be Chordata, the class will be Mammalia, and then we need to start to look at the diagram. So order here, we've got Carnivora, family is Felidae, genus then is Felis, and the species is Catus. So really early classification systems were just based on appearance, and there were issues with that because of what we call convergent evolution. So that's when 2 completely unrelated species have got similar challenges, and so they will adapt in the same way to overcome those particular challenges. So they call it convergent evolution when they converge. And some good examples of that is if we look at the dolphin and the shark, they're not closely related at all. The dolphin's a mammal, and the shark is a fish. It's a cartilaginous fish. So they're not closely related, but because they've got the same niche, they actually look very similar. So they've both got that sort of streamlined appearance. And they've both got pretty sharp teeth, and they've both got fins in very similar places. So they do have very similar appearance. So that's because of this convergent evolution. So we had to start looking at other things other than these physical characteristics in order to classify species. So the more modern approach then, we can use things like their physiology, so the way that they work, and also their biochemistry. And it was things like fungi where we weren't sure exactly where to fit them, what category to put them in, that made us start to think about these things in a slightly different way. So we've got lots of different molecules that we can actually look at. We can look at things like DNA, and we can look at different proteins. So all sorts of things. But what they want to do is to try and pick molecules that you'll find in lots of different species if we want to have a look at the evolutionary relationship or to classify particular organisms. So one of those things that they like to use is the cytochrome C. So cytochrome C then is a protein that's used in respiration. So all organisms are going to require this cytochrome C. But it varies from one species to the next. So this is where cytochrome C lives. It's in the membranes in the mitochondria, and you'll get more of an idea of the role of cytochrome C in the second year when we look at the process of respiration. But we're not really thinking about that. We're just thinking about how it can be used to classify organisms and look at evolutionary relationships. And they'll do that by actually looking at the amino acid sequence. So if they find that the sequence of amino acids are the same, then that means that the two species must be closely related to each other. And if they're different, then they're not closely related. And the more differences that there are between that amino acid sequence, the less closely related those two species are. So they can look at other proteins as well, things like hemoglobin, and you can see here by looking at hemoglobin, and this is the number of amino acids that are different. So gorillas here have got 1 amino acid difference from humans, rhesus monkey 8, mouse 27 different amino acids, chicken 45, and a frog 67. So we can see gorilla is the most closely related to us from looking at that. But we can also look at other things. So we can look at the DNA sequence. DNA is something that all living things contain, so it's quite a good thing for us to actually look at. And we can talk about this a little bit more when we look at evolution, but we can have changes in these base sequences, so this sequence of bases in the DNA, which are called mutations. You get random mutations. So what we can do is we can compare the DNA sequences in order to classify different species. And the more similar the sequence are, the more closely related two species are. And again, the more differences you have, the less closely related species are to each other. Now, what I wanted to have a look at very quickly was this idea of bioinformatics. So bioinformatics is what scientists would use in order to compare either their sequence of amino acids or sequence of bases. So bioinformatics then storage of biological data in databases, and then they can analyse that data and use it then to make sense of biology. So for instance, they can use genome sequences, they can look at single gene sequences, they can look at base sequences, they can look at amino acid sequences, they can look at protein structures. So all of those things in order to try and study that organism, look for evolutionary relationships and things like that between organisms. So I have done a demo for you. Here's my data. You can see I've got a lot of data, and actually continues off the page there. I can't show you that right now, but it goes on and on and on. So if I copy and paste that into this box here on the website, just there, and we make sure that we set it to DNA. So this website here, ebi.ac.uk tools MSA /Custoda. And then we just click on the submit button. What it does is it starts to sort that information for you. So you can see, actually, this is a bit neater there. We managed to sort it. And you can see actually that we had data for all of these different organisms. And then what we can do is we can click on phylogenetic tree, and it sorts that data for us and creates this phylogenetic tree for us, which is really clever. So we can see now how closely related various things are. So we've got the manatee here is pretty closely related to the African elephant, which is quite surprising. I thought they'd be more like cows, but there we go. We've got bottlenose dolphin in the killer whale, and a porpoise and a pig. So those are fairly closely related, which is also, again, quite interesting. And then we've got various other things. So we've got the sea otter and a polecat, a giant panda and a seal, fairly closely related. A rat and mouse, obviously no surprises there. So quite interesting things that we can do with this process of bioinformatics. Good. So now we're going to talk about the 3-domain system, which was actually based on DNA data, or actually RNA data. So they got RNA and they compared RNA, and they used to think that archaea and bacteria were really similar to each other. So they just bung them all together in the prokaryota, so they used to be in the same kingdom. But then they found out through looking at this RNA evidence that the archaea were fundamentally different to the bacteria, and in fact they had some characteristics that were very similar to bacteria, and they had other characteristics that are similar to eukarya, which actually changed our classification system. So it was very groundbreaking at the time. So paradigm shift. So all based on this RNA data. And we now know that bacteria are very different from both the archaea and the eukaryotes now. So they need to be in these separate domains. And they're different in a number of ways. So they've got a different cell membrane structure. They've got different flagella and different enzymes. They've got no proteins located on their genetic material, so there's naked DNA. Their DNA replication method is different. And archaea actually have many features that are very similar to eukaryotes. So they've got similar enzymes in order to build the RNA. That RNA polymerase is very similar. Similar method of DNA replication. Both have got proteins that are bound to DNA. So archaea are still classed as prokaryotic organisms because they don't have a nucleus. So we have got now two prokaryotic groups, one eukaryotic group in the domain system. If we look at the kingdoms, then we've got one prokaryotic group, and then all the rest of these are eukaryotic groups there. This originally was based on physical appearances, and I think because of all of these changes that we've got in the way that we're classifying organisms, and based on DNA evidence, that some of these things are going to get resorted fairly soon, especially this productista, which is the everything that doesn't quite belong in all of the other categories category. Yes, because so originally it was physical appearances. This domain system, however, then is based on modern methods. So this is because of the RNA evidence that they found. So it's a much more modern classification system. Good. If you've got any questions, then you can ask me via Teams or email me, and I will see you in future or listen to you in future. Bye.